Hello, everyone. I'm Cavan. I'm just going to try and give you an idea of how we try and work in the lab to look at the EEF1A2 mutations. So we use a range of techniques to try and look at EEF1A2. So my work is focusing on how do the mutations affect EEF1A2 function. And we use a range of different lab techniques. These involve looking at cells and the mice models that Grant has just talked about. But we also use computer programs to try and model the mutations. So how might this be useful? So we know that certain parts of the EEF1A2 protein are particularly important for its function. And so knowing where a mutation is and what it's nearby can be a really useful way to predict what effect the mutations might have. So we show you the EEF1A2 protein as the amino acid sequence here, but this is a bit of an oversimplification. As in cells, this string of amino acids is folded into a three-dimensional structure, like so. So if we know the structure, we can then model it using computational programs, and we would show the folded structure of EEF1A2 like so. And here I've just shown a little mutation, a mutated amino acid in red here, so you can see what it looks like. And what this means is that two amino acids that might not appear to be close together on the sequence, the amino acid sequence might actually be very close together. And so I'll give you an example. So here we have a mutated amino acid, so a mutant in red on the EEF1A2 protein. And in yellow, we have three amino acids that form a binding site. So they're a very important area for the EEF1A2 function. And we might not expect from the sequence that they're very close together, but when we fold the protein, we show that they're actually right next to each other. And so we might expect this mutation to be very disruptive for EEF1A2. And as you know, there are many different EEF1A2 mutations. And so by modeling where each one of them is, it can be a really useful way to predict what effect the mutation might have. So we can see which mutations are nearby to each other and which mutations are nearby to important parts of the protein. And this can be useful for guiding what lab experiments we carry out. And hopefully in the future, it'll be able to guide therapies and say which mutation, so which treatment might be most likely to be effective for each individual mutation. So just to recap, the function of EEF1A2, so as we've heard, the main job for this protein, the main job for EEF1A2 is to deliver the building blocks that make up new proteins. So without EEF1A2, cells aren't able to make new proteins. And these proteins are what makes things happen in the cell. So they carry out almost every major function. So it's a very important job. And what we don't know is how do the mutant forms of EEF1A2, how well are they able to make new proteins? Are they affected? And if they are affected, is this the same for each individual mutation? So one way we can look at this is through a technique called mass spectrometry. So if we imagine that EEF1A2 isn't doing its job properly, and its job is to make new proteins, we'd expect that maybe in the brain, we wouldn't see as many proteins, or the proteins that we do see might be different. So what we can do is we can take the brain of a mouse model, for example, the E12TK mouse model that Grant has just talked about, and we can run it through this technique, and we can detect and count all the proteins in the brain, and then we can see what has changed. And so you don't have to understand this plot, but each single dot is a protein in the brain. And so the ones that are really high up or far out to the side have changed more. And it's interesting to see, so this can give us a lot of information about how the mutation is affecting pathways in the brain. And you might recognize some of the proteins that have changed here that I've labeled, as they're often, some are involved in other developmental disorders. And that's because we do see a lot of overlap. So this can be really useful. And if we run this with different mutations, so brain samples from different mutations, and we see certain proteins are changing in both, it can be really useful for giving us targets for possible drug treatments and also markers so we can test these drugs in mice. So that's just an example of some of the ways we look at EEF1A2. Thanks very much. I hope you found this useful. Thanks to the organizers, and I'm happy to answer questions in the Q&A. Enjoy the rest of the conference.